Assalamu alaikum. So let's do the third part of the first topic, that is recombinant based molecular techniques. Um, this is really an exciting and new technology, and it's known as CRISPR-Cas9 system. In this system has and will revolutionize and change the field of not only molecular biology but also medicine as a whole. And in this system, it's a bacterial system that is composed of two components. A genetic system known as CRISPR, and an enzyme known as Cas9. Now, CRISPR stands for clustered, regularly interspersed, short, palindromic repeats. So let's look at it one by one word by another. So it's a repeat. So basically, it's a sequence that is repeated over and over again. And this sequence is palindromic. We have already defined what palindromic sequences are. They are short, they are interspaced, meaning that they are separated by other sequences, and this separation is regular. And overall, all of these repeats are clustered within one part of the genome. Okay, and this system is really created by bacteria, if I may say that, um, in order to protect themselves against bacteriophages or the viruses that infect these bacterial cells. Okay, Cas9 is a nuclease and it is RNA guided. So it's basically an ribonucleoprotein. Okay, so a ribonucleoprotein is a protein that is composed of a protein part and a nucleic acid part. Okay, so it is guided by this RNA sequence, and it goes into a DNA molecule that is complementary to this RNA sequence, and then the nuclease, the enzyme, cuts the DNA molecule that is complementary to this RNA. And this is known as, the RNA molecule is known as a gRNA, that is guide RNA, or single guide RNA, since it is single stranded. So what happens in nature is that whenever you have a bacteriophage that looks like this, and this bacteriophage infects a bacteria by inserting its genetic material into this bacterial cell, what the bacterial cell does is that it takes the bacteriophage DNA, it cuts it into different parts and then different fragments, and one of these fragments is taken and it is inserted in the CRISPR region. Let's say that you have another bacteriophage that infects the same bacterial cell. The same thing happens. The bacterial cell takes the bacteriophage DNA, it cuts it into different fragments, it takes another fragment and it inserts it into the same region, but it is separated, or the two bacteriophage DNA fragments are separated, so they are interspaced. Now, if you have a third one infecting bacterial cells, the same thing happens, and you have this portion of the bacteriophage DNA also inserted into the same region, and they are all interspaced regularly. Okay, so this is the CRISPR system. It's a genetic system that is part of the bacterial genome, bacterial chromosome, and it contains different fragments of different bacteriophages. Now, what happens is that if a bacteriophage comes in, if a bacteriophage that reinfects, if a bacteriophage reinfects the same bacterial cell, what the cell does is that it transcribes this region right here. So right here you have an RNA molecule that is produced. It gets processed. Notice that you have these are the palindromic sequences right here. Okay. Now the important thing about these palindromic sequences, since they are palindromic, that is, they are read from on one strand 5 prime to 3 prime, the same exact thing as the opposite complementary strand, also 5 prime to 3 prime. They will have this, the RNA will have this shape, and hydrogen bonds would form between the different palindromic sequences. Okay, so you have the RNA molecules looking like this. They get processed, and you have a CRISPR RNA molecule, or different CRISPR RNA molecules produced. Okay, what happens then is that each one of these RNA molecules is taken into the Cas9 nuclease. So this is the Cas9, and the RNA is inserted into it. So it becomes a ribonucleoprotein. Now, the Cas9 enzyme is guided by the RNA molecule. You have this complementary base pairing taking place between the bacteriophage DNA and the RNA of the RNA that is now part of the Cas9. And then Cas9, what it does is that it degrades the bacteriophage DNA. So now the bacteriophage DNA cannot overcome the bacterial cell, and this is how the bacterial cell can protect itself against this bacteriophage. So this is like an immune system of bacteria, which is quite sophisticated. So again, so what happens? You have a bacteriophage that infects bacteria. Bacteria degrades the bacteriophage DNA into different fragments. One such fragment is taken, it gets inserted into the bacterial genome, the bacterial chromosome, into a CRISPR array. So you have different fragments of different bacteriophages inserted into the bacterial chromosome. When you have infection of a bacteriophage into bacteria, the same bacterial cell, this system, the genetic system, is transcribed into one large piece of RNA. It gets degraded. It looks like this because it's palindromic. You have hydrogen bonds formed between within a palindromic sequence. So you have different fragments. These fragments, each one of them is combined with the Cas9 nuclease. The nuclease is guided by the RNA molecule. So you have a complementary base pairing taking place between the phage DNA if it reinfects the bacterial cell, and the RNA molecule that is produced from the CRISPR genetic system, and the nuclease degrades the bacteriophage DNA, and this is how bacterial cells can protect themselves against bacteriophages. Very nice system. Now, this system was actually discovered by two female scientists. I forgot their names. I should have their pictures here on the slide. And I believe that in a few years they will get the Nobel Prize for their discovery. Because what happens is that we can really take advantage of the CRISPR-Cas9 system by inserting an RNA molecule of our interest into the Cas9 system. So we guide the Cas9 nuclease into a region of interest, a region that would be complementary to the RNA molecule that is now part of the Cas9 protein. So we can really guide the Cas9 nuclease into a certain region, and we can cleave it, we can cut it. Okay, So what we can do is that we can create recombinant DNA molecules, plasmids that is, that contains the RNA, that contains the gene that would be transcribed into this guide RNA. Okay, and the plasmid would also produce Cas9 as well. Okay, since it is a bacterial gene, we introduce it into human cells. So the Cas9 would do exactly the same thing as it does in bacteria. That is, it goes into the human DNA that is complementary to the RNA. Okay, that is part of the Cas9 protein, and Cas9 would cut the human DNA at this particular site. Okay, so that's the whole idea. Now the thing is, how can we take advantage of this? Well, remember when we talked about DNA repair mechanisms, and we said that one such mechanism is when you have double strand breaks, and this is what Cas9 does. It creates double strand breaks. If you have double strand breaks in DNA, they can be repaired by recombinational repair mechanisms, a mechanism known as homologous recombination. So what it does is that it takes advantage of having of the cell being diploid, that is having two copies of every chromosome. So if one chromosome is damaged, what the cell does is that it takes advantage of the complementary chromosome or the homologous chromosome, and you can have exchange of genetic material filling in the gap created by the Cas9. So now you can repair the DNA, okay, using the homologous recombinational repair system. The other type of repair mechanism is known as non-homologous end joining. And what the cell does is that if it sees that you have double strand breaks, it glues the two strands with each other. Okay? Except that, so you can have the DNA now is continuous, except that the problem with the non-homologous end joining repair system is that mutations are created. So this gluing is really not perfect. So it's almost like if you have a vase and this vase is broken, well, you can glue the different pieces, but the vase would not really look exactly the same as before. Okay? So we really can take advantage of the CRISPR-Cas9 system by, for example, um, inserting the Cas9 and the guide RNA into these cells, the human cells, let's say. So the Cas9 is guided into a region of DNA that is complementary to the RNA. You have a cut that is introduced, double strand breaks, and these can be repaired again using the non-homologous end joining or the homology repair system. Now the thing is, if you have the non-homologous end joining activated and repairing the DNA, you can have mutations created. So you can have an insertion, you can have deletion of part of the DNA, or you can have frame shift mutation as well. And we talked about these mutations before, earlier. So what we're doing here is that if we guide the Cas9 into a specific gene, okay, and we want to disrupt this gene, we want to make it faulty, we want to disrupt it so that it doesn't become functional anymore, we can do this by activating the non-homologous end joining repair system. So we can create mutations so that the gene now is faulty, it's not functioning well anymore, and this is how we can actually study the importance of a gene biologically by disrupting it and seeing what happens afterwards. Okay? Now, the thing is with the homology repair system is that what we do is not only that we insert into these cells the Cas9 and the RNA molecule, the guide RNA, we also can insert a DNA piece that is homologous to the gene that we want to let's say, quote unquote, repair. So what we do, in fact, is that if you have double strand breaks in the chromosome and the homology repair system is activated, the repair system uses the homologous DNA that we have introduced by inserting it into the DNA. Okay? So what happens here is that we can truly repair our system. That is, let's say that here you have a mutation. What we do is that we insert a DNA piece that is homologous to that DNA, to the gene that is mutated. And what the homology repair system does is that it replaces the faulty gene with the new one, the correct one that does not have a mutation. So we can repair genes using this mechanism. So either mechanism, we can actually disrupt a gene. We can, let's say that this is a good gene and the homologous DNA that we introduce is mutated. Well, we can disrupt it either way, either that activating the non-homologous enjoining repair system, or by activating recombination, so that instead of having a fully functional gene, it's now exchanged by a faulty gene. Or if the gene is originally mutated, we can replace it by a gene that we introduce that is correct, that does not have a mutation. So in this mechanism, in the homology repair system mechanism, what we do is that we can replace a mutated gene by a normal one. And this is a huge, great opportunity of correcting genetic diseases by repairing DNA. Okay? Now, I found these images on Google and I loved them. I loved the images that is in this figure, for example, right here, we can manipulate genes manually. We can replace genes, we can cut them and paste them as we wish. This is tools, okay? مفتاح الإنجليزي مفتاح جرة الغاز. All right, so this here, you can use the similar tools. This is really the future, that is having DNA into a capsule, a medication capsule. This is the future. We introduce DNA into our cells, correcting mutated DNA pieces. Now, this is really extra. It's not part of the lecture. You don't have to study these, but just to show you the implications and the potential of the CRISPR-Cas9 system. That's a news piece in 2016. This is a scientist in the UK, and she applied for a grant. She wanted to get fund to use the CRISPR-Cas9 system in order to modify human embryos. And that was only for research purposes. So what she wanted to do was to disrupt and to disrupt the genes, okay, to see what their effect would be, okay, in human embryos, in fertilized eggs. And she didn't really want to continue with the fertilization. Okay? She only wanted to study what happens during the first 14 days. That was in 2016. And there was actually a controversy about this, because what would happen next? And actually, what happened next was, at the end of 2019, you have this happening. You have this Chinese scientist. What he did was that he really edited embryos, DNA of embryos. Okay, what he did, using the CRISPR-Cas9 system, what he did was that he disrupted a gene known as CCR5. So what's the significance of this gene? Well, CCR5 is it produces a protein that exists on the cell surface, and it helps HIV virus get into cells. So, if this gene is disrupted, then these individuals with mutated CCR5 cannot be infected by the HIV. So they are protected. So this is good, right? Well, the thing is also is that so, some research has shown that people with mutated faulty CCR5 are a bit more intelligent. Now, why? No one knows. Okay, but it has yet to be confirmed. So, what he did is that he modified this gene to make these embryos, to make these babies better human beings. Okay, so he got their brains enhanced. And these are the two girls that were born. And then he announced to the whole world what he did. Now, there was a lot of controversy about this, and so far, I think he has been jailed for 3 years. He has been suspended from his work for what he did. He thought that he was doing something really good to humanity, but he did something really bad. Why? Because one problem with the CRISPR-Cas9 system is that it is not really that accurate, meaning that it can go anywhere, somewhere in the human genome and it can disrupt it. So targeting is not really that accurate. It's not 100% accurate. And I read a piece of news actually yesterday um, about scientists in Australia showing that the CRISPR-Cas9 system is not really that accurate and it caused a lot of deletions in the human genome. But this is actually a start. That is, we can fix genes before any disease shows on any individual. So that's the potential of the CRISPR-Cas9 system. Okay, this is it. I wish you the best, and we will continue with the second topic later on.